Okay, thank you. Hello, today I'm here presenting on network anomalies during early recovery from alcohol dependence, a TMS-EEG network study. Before I begin, I just want to give a brief overview of the background of alcohol dependence. I realize my presentation is a little bit different to everyone else's. From a neurocircuitry perspective, the mesocorticolimbic dopaminergic circuitry plays a critical role in the development of addiction. Therefore, as you can see from the figure, a network of interconnected structures is implicated in the development and persistence of addictive behaviours. From a neurotransmitter perspective, ethanol, which is a psychoactive ingredient in alcoholic beverages, is a central nervous system depressant. Therefore, there is a delicate balance between the neurotransmitters following exposure to ethanol. Acute ethanol increases extracellular dopamine levels in the mesocorticolimbic circuitry indirectly through GABA and glutamate systems, while chronic alcohol actually induces a compensatory neuroadaptation to counter the acute effects of ethanol. Therefore, the delicate neurotransmitter balance required for adequate cortical inhibition, which is the neurophysiological mechanism by which GABA suppresses neuronal activity, is disrupted following chronic alcohol exposure. This imbalance has been extensively evaluated in preclinical studies. However, evidence from human studies is more limited as it's difficult to non-invasively examine cortical inhibition in humans. This is where we cue in TMS. As many of you know, cortical inhibition can be measured via application of TMS. In the past, TMS studies examined the effect of alcohol and found that ethanol had an effect on cortical excitability. However, these studies were largely confined to measuring cortical excitability of the motor cortex, as at the time, recording cortical inhibition from other regions was technically challenging. Combined TMS-EEG allowed researchers to measure cortical inhibition in the prefrontal cortex using a paired pulse TMS paradigm known as long interval cortical inhibition, or I'll call it LICI throughout. LICI has been found to quantify GABAergic activity in the frontal cortex, which is a region critical to the regulation of addictive behaviours. If you have a look at this figure, just if anyone's not familiar with it, following the single pulse in the blue trace, you can see the standard cortical response to the stimulation. When you apply the paired pulse paradigm, which means you apply the suprathreshold conditioning pulse prior to the test pulse, if you look at the red trace, you'll see that the cortical response is suppressed relative to the blue trace. And this is what we're interested in. So this led to the concept behind our earlier study, which examined cortical inhibition within the prefrontal cortex in alcohol-dependent patients during early recovery. For this study, we applied TMS-EEG to both the left and right dorsolateral prefrontal cortex of 12 alcohol-dependent patients post-detoxification and 14 healthy controls. 75 single pulses and 75 paired pulses were applied. LICI of the frontal electrodes were then quantified and compared between the groups. This was the first electrophysiological study to utilize TMS-EEG to quantify and demonstrate the presence of altered cortical inhibition in the bilateral prefrontal cortex of the alcohol-dependent group. So essentially what we saw was in the alcohol-dependent group, there was no suppression of the red trace, which is what we would see in the control group. So this was a really exciting proof of concept regarding altered cortical excitability of the frontal regions. However, as noted in the earlier slides, development of addiction recruits a network of structures. So I started to consider that perhaps we actually haven't properly utilized this rich data set, as it may be interesting to also examine global features of brain connectivity, which is essentially how these structures communicate between them as a network. And the reason I was particularly excited about returning to this data set is that we had elicited essentially two different brain states, whereby the paired pulse TMS perturbation elicited a disrupted inhibitory network brain state, which could be compared to the single pulse state. This provides us with a really unique opportunity to map the network connectivity when in an altered state. And so this was really exciting for me. So just as a very quick overview, I know I'm throwing a lot of concepts out there. What is a network? The network analysis is a brain mapping technique which is used to quantify network connectivity within the interconnected brain. Using this analysis approach via graph theory, researchers are able to actually extract topological features of brain connectivity. So this includes looking at network features such as global structure, global integration, and global efficiency. So using the data from the earlier study, we could now start to examine the brain networks underlying alcohol dependence in early recovery and provide a characterization of the global network response to the single impaired pulse perturbation when TMS is applied to the frontal cortex. So this led to our more recent paper, and this paper is currently being under revision, so hopefully it'll be published soon. So what I'm showing you is really a sneak peek of this publication. Recently, studies have examined the networks underlying acute and chronic alcohol consumption. It's very early days for these studies. The majority of studies conducted are fMRI and DTI, which have generally observed a reduction in global efficiency and altered integration associated with chronic alcohol exposure. EEG studies are far more scarce. Acute exposure was found to be associated with increased global efficiency, while EEG findings regarding chronic exposure are far more mixed. However, taken all together, there seems to be an emerging pattern of an altered network associated with chronic alcohol use. In the current study, we transformed EEG signals to a correlation matrix using pairwise Pearson correlations. A correlation matrix was constructed for each condition, and these matrices were constructed for each participant. From each correlation matrix, network measures were extracted, such as mean degree, clustering coefficient, and global efficiency. Our main findings were that, firstly, very interestingly, there were no network differences in response to the single pulse, which is what I had expected, but it was a really nice proof of concept. Okay, I'll go faster. However, following inhibitory paired pulse TMS to the left dorsolateral prefrontal cortex, the alcohol-dependent group exhibited an altered network response. So global efficiency, which is a measure of information transfer, was significantly reduced. Additionally, using predictive modeling, we found that reduced global efficiency was predictive of being in the alcohol-dependent group. We also found that the alcohol-dependent group presented with significantly reduced mean degree, which reflects the processing of information and communication within the network, and reduced clustering coefficient, which is a measure of locally specialized connectivity. Both reduced mean degree and decreases in clustering coefficient were also both strongly associated with increased severity of past alcohol use. We also examined whether the network response correlated with the ERP response, and we found it did not correlate. Therefore, it appears we are tapping into two different aspects of local and global functions of the brain across these two studies. So therefore, our findings identified altered networks which persist in early recovery and noted a relationship between altered network features and clinical aspects of alcohol dependence. This is really exciting and a new approach for looking at addictive behaviours, and we propose that this novel direction could potentially assist in identifying biomarkers of alcohol dependence and relapse. And I believe it can really contribute to brain mapping tools and therapeutic tools for addiction by using brain stimulation techniques. So finally, I'd just like to say thank you to all my co-authors in helping me shift these theoretical thoughts into experimental studies, pilot studies. Thank you for listening, and please feel free to contact me if you have any questions. Thank you.
Thank you, Jodie. We don't have very much time for questions, unfortunately, but there is one. Great talk, Jodie. There is a lot of evidence suggesting later TMS-evoked EEG components may reflect sensory potentials despite auditory masking. Do you think this could have affected your data?
It's a longer answer than the question allows me. Yeah, it's an interesting point. We use white noise to mask like auditory noise from the click of the TMS. But that's something really interesting to look at, and maybe I can look at the localized global response if they're talking about the network response, and we can actually locate where we're seeing the differences. But for the first study, we were looking at the prefrontal cortex, so I think it's more likely to affect other electrodes. So we'd say dominate there. Yeah, very quick answer. So you can email me after if anyone wants to ask more about this. Thank you.
Great presentation, Jodie. I don't think 8 minutes was enough to do 2 studies. Well done.